Bone ossification refers to the formation of bone tissue. So making of bone, so that's what this video will be about. Ossification, again, the formation of bone, occurs in humans about 8 weeks post-conception. So in utero, a developing fetus would go through ossification. In this image here, the dark patches in this skeleton are primary ossification centers, locations where bone tissue is forming. So what is the rest of the skeleton at this point? It's cartilage. A lot of the skeleton in an early developing baby is cartilage, which then turns into bone throughout development. That is one way that bone is formed. So the two mechanisms of bone formation, one is endochondral ossification. This endochondral refers to inside cartilage. So this is a cartilage replacement model where bone is taking over where cartilage once was. This is occurring in most of the skeleton, and all of these locations you're seeing throughout the long bones, for example. All the bones besides the skull and the clavicle. The skull and clavicle, those are examples of flat bones that go through intramembranous ossification. What this means is inside of a membrane, bone tissue is forming. So this is bone formation without cartilage replacement. Instead, stem cells are dividing and turning directly, immediately into osteoblasts to produce bone tissue without a pre-existing cartilage. So we'll talk a little bit more about endochondral ossification occurs more. Both these mechanisms are actually important for bone growth as well. So it's important to briefly cover both mechanisms. Just to review a bit of anatomy that you should have seen in a previous video, I want you to remember the term epiphysis, that are the ends of bones, and then the diaphysis is the long shaft of the bone, medullary cavity in the middle, and then I briefly mentioned the epiphyseal line before. In a mature adult bone that's done growing, done lengthening even, besides some remodeling still going to happen, there is something called an epiphyseal line that is the remnant, the visible remnant of an epiphyseal plate. Epiphyseal plates are where long bone growth occurs. So just want you to know that anatomy that we'll use a little bit in this video, and then in the bone growth video, you should know about the epiphyseal plate. One more thing to review before we jump into the bone growth. You may remember differential gene expression. I hope you do. So here is an egg being fertilized by a sperm. Those cells go through mitosis and divide, and then those cells start to specialize or differentiate to have different functions. The very simple schematic of that is they differentiate into 4 different tissue types. In reality, you're starting to learn now that each tissue type has more than one tissue type. So connective tissues are very diverse in themselves, for example. So of course it's more complicated than this. So here's a little bit of a view of some of the complexity. Embryonic stem cells would be these early cells that are able to differentiate into anything still. And that's what is shown here. So there's these 3 broad categories that later differentiate into tissue types: the mesoderm, endoderm, and ectoderm. Mesoderm is the one I want to refer to briefly right here, and specifically one of the substances it differentiates into is the mesenchyme. Mesenchyme is an embryonic connective tissue. All connective tissues, as well as smooth muscle and cardiac muscle, differentiate from and derive from mesenchyme. So mesoderm is going to differentiate into mesenchyme. It's a further specialization. Tubules of the kidney are going to differentiate from mesoderm into something else. It's not mesenchyme. So mesenchyme is going to be an early connective tissue that then can differentiate into chondrocytes, fibrocytes, smooth muscle, cardiac muscle, osteocytes, etc. So with that in mind, let's talk about what mesenchyme does here. So this is intramembranous ossification. How this occurs is it's going to start with the mesenchyme, which is right here's a mesenchyme cell. This is the mesenchyme. So it's early specialized cells that have differentiated from mesoderm. It's going to form a membrane. This is what the intramembranous refers to, this inside this membrane here. Osteoblasts are going to line up along the blood vessel. So the mesenchyme is going to develop into osteoblasts, right? That's what mesenchyme does. It's a stem cell. It's able to divide and differentiate into other cells, for example, osteoblasts. So osteoblasts have now differentiated here, and they're going to produce osteoid. Osteoid is the unmineralized organic portion of bone. Wait, what is that? That's collagen. That's the ground substance without the calcium. So that means that it's a lot like hyaline cartilage, but it's not, because we have osteoblasts, but it's similar in structure. It is immature bone tissue. So then we've got everything here. Next, what's going to happen is we're going to have calcification, right? To get to bone, we have to calcify that matrix. Calcium phosphate is deposited, so the tissue is mineralized. Osteoblasts mature into osteoclasts as they're trapped in this matrix. So these are now osteocytes here in the middle. We still have osteoblasts out here because we're still forming bone tissue. We still need osteoblasts to be blasting out more collagen. But osteocytes are also here now. Those are mature bone cells that have become trapped in that hardened matrix, and they have made themselves a little cavity called a lacuna to live in. We have trabecula starting, so those kind of holes in spongy bone that are starting to be the formation of actual bone tissue opposed to that osteoid tissue only. Okay, next we've got the mesenchyme forming the periosteum. Oh, that's a fun thing to say. Mesenchyme forms the periosteum. Remember, mesenchyme is a precursor to many connective tissue types. So it can differentiate into fibroblasts that make this dense connective tissue that surrounds the bone, the periosteum, fibrous lining of bone. In here, we've got calcified trabeculae. That is our spongy bone. We now have a honeycomb of spongy bone in the middle here. And we kind of keep going here. Lastly here, we've got osteoblasts at the surfaces that are still present, are going to be filling in the gaps between the trabeculae. This is going to make us go from spongy bone to compact bone on those edges right next to the periosteum. So as those osteoblasts produce and blast out a bunch of collagen, that makes the bone more compact. Of course, calcium's coming in to harden that. This results in this sandwich-like feature, a flat bone. Remember, these are flat bones that are going through this type of ossification. We've got the compact bone, spongy in the middle, compact bone on the other end, sandwich with spongy in the middle. And that's a flat bone for you. Okay, next up is endochondral ossification. This is the cartilage replacement model. So we're starting with cartilage. So this bone here is, I'm calling it a bone, but is it bone? Not really. It is cartilage, hyaline cartilage, and it does have a bone collar here. So we're starting with hyaline cartilage, and you can follow across development here if you'd like. This is week 9 of post-conception, right? So still in utero. So the whole thing with this is enlarging and dividing chondrocytes that then deteriorate and are replaced by osteoblasts. So let's just walk through this. In the middle here, we've started to have deteriorating cartilage. How did this get longer, this here? Well, we had cartilage dividing. I actually want to go to this nice image I have right away here. So this is the process that's happening throughout this entire thing here. On top, we've got chondrocytes that are dividing and enlarging. They're growing bigger, they're dividing, and that's adding length onto the end. Remember, these are long bones. Onto the ends, those epiphyses are getting longer because we have cartilage dividing, chondrocytes dividing. At the bottom, we've got chondrocytes that start to degenerate or degrade. So that's kind of happening right here. We'd have degradation. Up here, we have dividing. As those chondrocytes degenerate, osteoblasts come in there and invade and make bone matrix, because that's what osteoblasts do, right? So they're kind of moving, pushing up, taking over those dead chondrocytes. So moving up here, what that results in the middle here is the formation, start of a medullary cavity. Now that we have deteriorated, like a hole here where the chondrocytes degenerated, we can now have blood vessels invade. Remember, bone tissue is vascular, so there are blood vessels in bone. There's not in cartilage, so this helps the spongy bone form. This is called our primary ossification center. Actually, I have it labeled over here. This middle section here, primary ossification center for endochondral ossification. This is what's happening at that center. This same process here is happening at the secondary ossification center, as well as at the epiphyseal plate for linear bone growth. So understanding this process is really important. So this is that primary ossification center that's forming the cavity, the medullary cavity, as well as adding bone matrix to this end as it pushes up, as well as adding cartilage as those cells, chondrocytes, divide on the ends of these bones here. So as the collagen thickens, as we have these osteoblasts migrate upwards and replace the bone, the osteoblasts are laying down the bone matrix on top of that cartilage base. What's that cartilage base? Collagen, right? We've already got the collagen base, some ground substance. We're adding new collagen from the osteoblasts, as well as bringing calcium phosphate in. So as the new bone is added at the ends, the shaft lengthens. The other thing we have here is the secondary ossification centers at the epiphyses. So the epiphyses are going to ossify kind of separately, but the same mechanism is occurring here at the secondary ossification centers, one at each end, each epiphysis. And then what's going to happen is the epiphyseal plate will remain here, and this is still cartilage. This is going to allow this same mechanism to happen and allow for linear bone growth, again, by these chondrocytes dividing on one side, lengthening the bone by dividing, and then the bottom of them deteriorating and bone taking over, pushing up underneath, intruding where those chondrocytes were. But it's really the chondrocytes dividing that add the initial length to the bone. All right. So what we've done here is compare and contrast intramembranous and endochondral ossification, briefly describing each process. So the difference between the two, I want you to briefly know, as well as describe each using these terms here.